In Star Trek, the Borg are a ruthless, hive-minded collective that assimilate other beings with the intent of taking over the galaxy. Here on non-fictional planet Earth, Borgs are DNA packages that could help humans fight climate change. Last year, a team led by Jill Banfield discovered DNA structures within a methane-consuming microbe called methanopridans that appear to supercharge the organism's metabolic rate. They named the genetic elements borgs, because the DNA within them contains genes assimilated from many organisms. Yet they play an outsized role in Earth system processes by removing methane, the most potent greenhouse gas, from the atmosphere. The gas is emitted naturally through geological processes and by methane-generating archaea. However, industrial processes are releasing stored methane back into the atmosphere in worrying quantities. As part of this work, she and her colleagues regularly sample microbes in different habitats to see what interesting genes microbes are using for survival, and how these genes might affect global cycles of key elements, such as carbon, nitrogen, and sulfur. The team looks at the genomes within cells as well as the portable packets of DNA known as extra-chromosomal elements, ECEs, that transfer genes between bacteria, archaea, and viruses. These elements allow microbes to quickly gain beneficial genes from their neighbors, including those that are only distantly related. Using advanced genome analysis tools, the scientists determined that many of the sequences within the Borgs are similar to the methane metabolizing genes within the actual methanopridans genome. Imagine a single cell that has the ability to consume methane. Now you add genetic elements within that cell that can consume methane in parallel and also add genetic elements that give the cell higher capacity. It basically creates a condition for methane consumption on steroids, if you will, explained co-author Kenneth Williams, a senior scientist and Banfield's colleague in Berkeley Lab's Earth and Environmental Sciences area. The East River Field Site is part of the Department of Energy's Watershed Function Scientific Focus Area, a multidisciplinary research project led by Berkeley Lab that aims to link microbiology and biochemistry with hydrology and climate science. Our expertise is bringing together what are often thought of and treated as completely disparate fields of inquiry. Big science that links everything from genes all the way up to watershed and atmospheric processes. Resistance is futile, a disadvantage Banfield and her fellow researchers at UCB. Based on the similarities in sequences, the engulfed cell could have been a relative of methanopridans. But the overall diversity of genes found in the Borgs indicates that these DNA packages were assimilated from a wide range of organisms. No matter the origin, it is clear that Borgs have existed alongside these archaea, shuttling genes back and forth, for a very long time. Notably, some methanopridans were found with no Borgs. And, in addition to recognizable genes, the Borgs also contain unique genes encoding other metabolic proteins, membrane proteins, and extracellular proteins almost certainly involved in electron conduction required for energy generation, as well as other proteins that have unknown effects on their H. Until the scientists can culture methanopridans in a laboratory environment, they won't know for sure what capabilities the different Borgs confer, why some microbes use them, and why others don't. One likely explanation is that Borgs act as a storage locker for metabolic genes that are only needed at certain times. There is evidence that different types of Borgs sometimes coexist in the same host methanopridine cell. This opens the possibility that Borgs could be spreading genes across lineages, said Banfield. Some researchers are combing through data sets of genetic material from other microorganisms, looking for evidence that Borgs exist in association with other species. while her colleagues are using lab-based methods. Co-author Susan Mullen, a graduate student in Banfield's lab, will be getting her feet wet with some very picturesque fieldwork. She recently started a project to sample microbes from the floodplains of the East River throughout the year to assess how seasonal changes in borg abundance and other microbes known to be involved in methane cycling correlate to seasonal fluxes of methane. According to the authors, years down the line, carefully cultured microbes chock full of borgs could be used to reduce methane and curb global warming. It's all to benefit the collective, life on Earth. https://ift. pjviarl. Accessed October 19, 2022. Thanks for watching. Please subscribe my channel for more videos.